Genetics is the study of inheritance and is about the transmission of our genetic information, or our DNA, from one generation to another. That genetic code contains information about the genes that are going to be expressed in all our different cell types. Epigenetics literally means epi, on top of, from the Greek, genetics. And these are chemical modifications or marks that sit on top of our DNA or on the proteins around which the DNA is wound that influence how that DNA is packaged. Because in fact, in all our cells, we have 2 to 3 meters of DNA, and that has to be packaged into the nucleus of a cell, which is of about a 6 micrometer diameter. So we've got lots of that double strand, that helical DNA that has to be packaged into the nucleus of a cell. That DNA is wound around histone proteins, which form chromatin, which is then packaged into the chromosomes, the bundles that sit within the which harbor the genetic information that sits within the nucleus of our cells. The types of modifications that influence how DNA is packaged comes in 2 forms. Modifications to DNA, such as DNA methylation, that sits directly on the bases, on the code of our DNA, and also chemical changes, chemical additions to the tails of the proteins, the histone proteins that form the nucleosome around which that DNA is wound. And that packaging influences whether a gene is turned on or a gene is turned off. So while the nuclei of our cells, of all our cells, all contains pretty much the same DNA, our different cell types have different epigenetic modifications. So genomes and epigenomes are different from each other. So a liver cell will need to express genes that are important for making a liver function, and the packaging of the DNA in a liver cell enables those genes to be expressed. Whereas in a brain cell, a neuron, brain-specific genes need to be expressed. Those liver genes are tightly packaged, so they can't be turned on. And instead, you have open chromatin that enables genes that are important for neurons to be expressed. So my research is very fundamental research. I'm not doing translational research or very applied research. My research is trying to make conceptual advances in how the epigenetic modifications regulate gene expression, genome function. And I do that in 2 areas. One is I'm particularly interested in the dark matter of our genome. So our DNA is made up of sequences that code for genes, but actually that's just a tiny, tiny fraction of our DNA. Only about 1% of our genome is encodes genes. The rest of it encodes regulatory sequences that are important for whether these genes are turned on or off, and those are epigenetically controlled. But also about half of the human genome comprises repetitive sequences that are repeated over and over again, many of which look like viruses, and many of which have the potential to jump around within our genome, which makes our genome quite vulnerable. And one of the things that epigenetic modifications has evolved to do is to target those repetitive sequences and make sure that they are invisible to the machinery of the cell that turns genes on and off. So I'm very interested in how those epigenetic marks are targeted to these repetitive sequences. The second area that I use really as a model for studying the epigenetic control of genome function is a process called genomic imprinting, which is a process that causes genes to be expressed from only one of the 2 parental chromosomes. So for example, there's a growth factor gene called insulin-like growth factor 2, which is very important for the growth of the embryo. And that is only expressed from the copy that is inherited from the father. The mother's copy is there in our DNA, but it's epigenetically modified so that the transcriptional machinery, the machinery that turns genes on and off, can't see it. and that only the paternally expressed one is being made. And that's important because if by mistake the maternal copy gets turned on, you have an extra dose of this growth factor, and that has consequences for the growth of the fetus. You get overgrowth and actually an increased incidence of childhood cancers. So the dosage of imprinted genes is very important, and it's epigenetically regulated. So we've been studying this process of how the machinery that turns genes on and off tells the difference between the two parental chromosomes at these imprinted genes. And actually, they're quite large regions of the genome that are regulated in this way. And we're trying to understand the architecture of these chromosomal regions and all the features that enable parental origin-specific gene expression. And through understanding that mechanism, we can then apply it to the epigenetic control of other genes that are not regulated by imprinting. So one of the things that particularly excites me about the study of these two areas is the opportunity to understand something called epigenetic inheritance. Now, epigenetic inheritance is a more controversial area because it's addressing the question of whether epigenetic information can be transmitted from one generation to the next. Now, genetic information we know is the template, is the blueprint of heritability. But the question really is, can epigenetic marks also contribute in that heritable way? Now, the thing about genomic imprinting is that we have two parental chromosomes, one set that's inherited from mum, one set of chromosomes that's inherited from dad. And I've told you that genomic imprinting causes genes to be expressed from only one of those two parental chromosomes. And it's always the same one. So it's always the copy that's inherited, the copy of the insulin-like growth factor 2 gene that is inherited from father that is expressed. The mother's is always silent. That means in the developing embryo, there needs to be a memory of whether that chromosome originally came from the mother or came from the father. In other words, that is a model, a potential model for studying epigenetic inheritance, because that epigenetic mechanism has to have come from the egg or from the sperm, because it's marking those two parental chromosomes as being different. These are not genetically different. The genomes of males and females are not identical, but the epigenetic marks at these two parental chromosomes are very different at imprinted loci. How can there be a memory of parental origin that is transmitted from parent to offspring in a mechanism of epigenetic inheritance? And we've been studying that, and we now understand the mechanism and the proteins that are required for that maintenance. And what our work has shown is that the key time for that memory, which is placed in the egg and in the sperm, is actually that milieu of the egg immediately after fertilization, when the rest of the epigenome is being erased. Is being erased and reconstructed to make the embryo. Epigenetic marks are very dynamic. But this imprint memory has to be resistant to that dynamic change that's going on in the early embryo. And there's a set of proteins, two proteins actually, that are required for making sure that that memory is retained in an environment where everything else is being lost. One of the reasons why the study of epigenetic inheritance is controversial is because we know that epigenetic marks immediately after fertilization become erased in early development and reconstructed. And that's so new marks can be placed to regulate genes that are very important for early development. So that reprogramming, that erasure and reconstruction of our epigenome that's happening on top of our stable genome is really important for normal development. Now, the thing about genomic imprints is that they have to be a memory of the maternal and paternal chromosomes, and therefore they cannot be erased and reconstructed, otherwise the chromosomes would lose their memory. The mechanism of inheritance that we all know and understand is through our DNA, is through our genetic material. So the idea that something that is not genetic, that is epigenetic, might be inherited is quite controversial. So the idea that epigenetic marks could be inherited from one generation to the next is a really important and challenging and difficult question. It's a very difficult question to study experimentally, because a lot of the epigenetic variation that one sees in one's genome is actually due to underlying genetic changes or genetic variation. But it's important because there is a growing thinking within the field that the environment talks to the genome via the epigenome, and that while our genome is stable, our epigenome, which is more flexible, could be the mediator of environmental information, which places a new perspective on evolution, actually, and on the classical dogma about inheritance only being based within our genetic information. Now, which is why we study models of epigenetic inheritance, true models of epigenetic inheritance, like genomic imprinting. And what the studies on genomic imprinting have told us is that in order to get that memory, that epigenetic memory from parent to offspring at an imprint, that is a highly regulated process that has evolved through the function of these two proteins that are required for maintaining that mark when everything else is being erased. To get epigenetic inheritance is asking a lot. As I told you, the epigenome is erased and reconstructed immediately after fertilization. So for a mark to be heritable, it would have to be refractory to that erasure process.